I'm ready.
Okay, so we'll welcome our third speaker in this session. So Nima Darafshi is going to speak to us today about unraveling the mystery of prey tracking in bearded dragons, a study in smooth pursuit and distance.
All right. Hello, everyone. Well, my name is Nima. Thank you all so much for being able to come. This is my entire Steinmetz presentation that's been advised by Professor Fleischman. Here I had two different lizards. One of them is Flicker with some bait on his head, and then one of them is Helios with his little tongue out posing for the camera. So let's get started. So, what is my experiment? When we did a lot of in-depth research and getting information about the behavior and especially the visualization of bearded dragons, we especially saw in general with lizards that they primarily do not utilize smooth tracking. Chameleons, in fact, are probably the most prime example where they have one eye that's able to track and find different food that's looking around and then shoot their tongue out to get their bait. But when we were looking at bearded dragons with our records and especially videos, we observed something pretty differently. And we wanted to accurately track if bearded dragons, and if they are able to, see and utilize smooth tracking with their bait. So right here is a little setup where all the magic happens with a light and a camera to point down and see what we are looking at. And using my mastered self-taught drawing right here, we have 3 points of reference. One of them was right at the back of the head for our lizard that we made with markers. Not permanent Sharpie markers, more Crayola ones. One right at the tip of the nose right here for our head assessment. And then our third point was tracking the cricket food or the lizard itself. And these 3 points of reference was able to be computed into our MATLAB software that we ran to especially figure out and test what is the alignment between all 3 of these points and calculate what is the error of the head alignment compared to the bait as it moves and oscillates back and forth. So a real example of this, we can actually be able to see in this top left corner. This is a pretty solid alignment we have here. And then in this bottom left, we can see a little bit of an error. But more importantly, when we were swinging the bait back and forth, we were actually able to see that his head is moving side to side, back and forth to make sure that it is ready before it jumps and shoots out to attack. So, getting into the results, there's a lot of data and a lot of different graphs up here, so I'll try to break it down as easy as possible. The videos that we used were shot in 60 frames per second, and all of our x-axis shows the frame. And the frame is individually one by one. And our y-axis has been plugged into different softwares to understand and calculate and translate all of that into millimeters of how off and how inaccurate was the head alignment to the bait as it was swinging back and forth. And for a point of reference, whether it was a cricket or it was bearded dragon food, we were able to have a good margin of error of 5 millimeters in terms of the width for each of that food. So any information and any data that we got that was under 5 millimeters for alignment is pretty damning of information to show that these bearded dragons are lining up almost perfect with the tip of their head to the back of their head. And we can actually be able to see that the mean for Flicker was 2.69. So he's our golden child and kind of our prodigy when he's trying to keep up and move his head with the bait and the pathway as it moves back and forth. This top left graph right here is coordinates that we use to track the movement of the head as it's moving in the exact same direction and sine Hilly wavelengths of the bait as it's swinging back and forth. And to see that we're having a mean of 2.69 millimeters is almost perfect. And we can see that with our alignment and also the pathways of the head and the bait. Helios, on the other hand, he's trying, but more importantly, we're able to see, especially disparities within our data too. The head path movement, since it's pretty flatline and stagnant, that tells us that Helios is not moving his head at all. And in comparison to the bait as it oscillates back and forth, we can see in our alignment score itself that it's matching up perfectly with the bait movement too. And the mean among this is 7.58, which shows that there is some inaccuracy with Helios as he's moving his head back and forth. But once he's able to catch and lock on to that bait swinging, the data is at well below 5 millimeters for him to be very accurate and keep on track with the bait. So we had two eyes that we were able to look at, but more importantly, we wanted to know what would happen if we covered one eye to really test the exact perception as they're trying to find this bait too. And what we were able to realize is not only are they not able to keep in a perfect pathway and cycle, they have a lot of stutters and especially a lot more stagging head movements as the bait is swinging in front of their head back and forth. So when we were able to graph all of that data, we were actually able to see that the mean for flicker with their left eye was 6.77 millimeters, which showed a little bit more inaccuracy. And we can even see that in the head pathway as well. While it is almost a perfect alignment with the bait, it is still very, very rough and scattered all over the place, which is still identifying that we are having a lot of data above this 5 millimeter threshold, which is showing that when you cover up one eye, there is going to be some inaccuracies with the left. And for flicker, even though they're our golden child, our prodigy, the right doesn't get a lot better. The head pathway is not able to move as continuously as the bait, and the mean for that is 15.45 millimeters among all of our trials. And one of the important things we wanted to make sure that these lizards can be able to refine and make sure our results are airtight is to get them accustomed to the tape that we're putting over their eyes so that stress is not another level that we aren't accounting for. We want to make sure this data is as accurate and as perfect as possible. And what we can see when we use two eyes versus left or right, they're being more inaccurate when they have an eye covered. Looking off with Helios, there is some pretty solid and good data that is moving back and forth. However, the mean is still 7.12 millimeters, which is showing room for inaccuracy, especially when we're looking between the 200 to 300 frames. And then when we look over to the right eye, it's a little consistent, but it's still a little bit all over the place when we are looking at our alignment graphs, and our mean for all trials is 9.78 millimeters too. And then what we wanted to do is especially compare and contrast both the left, the right, and both eyes as well between flicker and Helios. Flicker has the blue data, and then Helios has the orange data, that we were able to calculate what is the distance off from the perfect alignment and compare that between all the different kinds of experiments we ran. For flicker, our golden child, we were able to see that both eyes are very accurate below that 5 millimeter threshold, while the left eye gets a bit more increasing and the right eye even more. And especially the bars that we had were standard deviations, and if there aren't many scientists in the room, standard deviations basically tell us how scattered all of our results were and what the mean was in terms of the alignment among every single test that we did. So especially with flicker, the right eye is very all over the place. Sometimes they were pretty accurate, sometimes they were very off with their alignment. Same with the left eye too, but with both eyes it looks pretty solid, and sometimes there are anomalies with Helios's data as well. But alignment is not the only thing that contributes to a bearded dragon's ability to use their ambush predator instincts and hunt for different kinds of food. A lot of that is also distance perception, and also being able to track how far away the bait is so they can shoot their tongue out and be able to get that little piece of bait right in front of them. And this introduced two different ideas of vision that is important for bearded dragons. One of them is stereopsis, and that is the ability to perceive a 3D structure's depth using different lines of sight. One of the best ways to understand this is by looking at bait, and the usage of stereopsis basically tells the species and the individual how far away your object is from your initial standpoint using both of your eyes. And that's the key thing we need to know. That you need to use two eyes for this kind of assessment. When we're looking at focus, while focus doesn't really utilize it for depth, focus is very important to make sure that the 3D structures it's looking at is very clear and sharp, and that what they're looking at is a very accurate bait that they can be able to lunge for. And while they can be able to use two eyes, one eye is still very, very satisfactory to make sure they can be able to get their bait. So different hypotheses we had for this is that with the distance experiment and what we were going to track when they start to protrude their tongue before getting their bait, one of them is that if the bearded dragons are using focus, then we're not really going to see a difference in when they start to shoot out their tongue with 2 eyes, 1 eye, or the other eye. But if bearded dragons use stereopsis, we will see a difference in the data when it's covered or uncovered with all of their eyes. So moving on to the data for this, we actually see that there is pretty similar data for flicker, almost about 1 centimeter off from the head movement to the bait itself when we're comparing the left, the right, and both eyes. And again, these bars are showing our standard deviation. However, there isn't as much of a huge disparity among all of the means that we have right here among left, right, and both eyes. And when we look at Helios, he was pretty perfect for every single attempt for both eyes, and the difference he has is about 2 centimeters, and even then, it's about half a centimeter from both eyes to left and right. But even then, there isn't huge disparity that we're noticing with their tongue protruding before they get their bait. Although, even though the graphs show something different, what we were actually able to understand and observe is that when they have 2 eyes, they're able to shoot straight forward and get their bait. However, if they do have 1 eye covered, they usually attack at an angle. And that was something interesting that we were able to look at. And even better is to kind of show the videos that are in this. With Helios right here, even though he's not a star player, he shoots straight at it to go and get the bait. While if you cover up 1 eye, he goes for a few attempts, shoots a lot more blanks, and is really trying his best. And this doesn't really go on for a few seconds. This actually goes on for about 30 seconds until he's eventually able to finally get the bait at the end. So, what does all of this data really mean with our alignment test and also our distance test? Well, one thing we really noticed, which was kind of a scientific breakthrough, in our alignment test, we saw that bearded dragons can smoothly track fast-moving objects and bait as it's swinging in front of their face, and that their head alignment is much better without covering their eye. So we understood that their margin of error from perfect alignment is a lot less using both eyes compared to a left or a right. And then we can be able to understand that their head alignment utilizes stereopsis to understand what the depth is and how they can be able to keep locked on and on track with the depth as it's moving back and forth for their bait. Meanwhile, for distance, we kind of had 2 interesting observations. Well, one of them is that their distance judgment is not really off when you have both eyes versus 1 eye, when they start to protrude their tongue and jump to get their bait. However, the angle of attack is impaired when we do cover 1 eye, whether they're shooting at a curved left angle or shooting at a curved right angle. So we were able to understand and say that the distance clarity, when they're able to perfectly align resolution within their bait, that is utilizing a lot more focus, pretty similar to focusing on making better resolution and clarity. But the angle of attack at which they try to jump and ambush their prey is actually based on 2 eyes, which we can see when an eye is covered up. And that is really damning evidence for stereopsis. That's kind of the end of my presentation. I wanted to thank the biology department for all the lab facilities that I had, for all the professors, whether they taught me molecular biology, herpetology, behavior, whatever it may be, and especially thank my advisor, Professor Leo Fleischman, for answering a lot of my questions, making sure that when I started with a whole new project in the winter, it went as smoothly as possible. But any questions or comments? Thank you.
Yes, if I know Nima, he would be more than happy to answer questions.
Is there any evidence for eye dominance in this organism? And if so, how might that affect the interpretation?
What do you mean eye dominance? Like is it left eye?
When you have 2 eyes that are working, at least in us, and presumably in a lot of animals, there's 1 eye that's dominant in the other. Even though we've got stereo vision, there's 1 eye that's more dominant than the other in terms of presumably how it's perceived in the brain and all that stuff. So I guess the question is there any evidence for that in these lizards? And if the answer is yes, how might that affect your interpretation of some of the data?
Yeah, I think that might actually be circumstantial from eye to eye. When we were able to look at the distance data for Helios and Flickr, they were pretty similar, but their alignment more specifically. We saw with Helios, he had 9.78 and then 7.12, while Flickr had 15.54 millimeters versus 6.77. While I wasn't really focusing more on right or left, that can be a really interesting topic for another future junior to pick up that study. But in terms of saying that one eye might be a bit more dominant than the other, I'm not 100% sure. What we know is that both different individual eyes are weaker compared to the uncovered eyes too. Any other questions or comments or compliments?
Compliments.
Great job. Thank you. I also had a question. So, your data suggests that the bearded dragons do have smooth eye tracking, which is dissimilar to other lizard species. I think that was the introduction.
Yes.
Do you have any idea why, based on perhaps established research, why bearded dragons are sort of special? And are there other lizards that are also sort of in this special category like bearded dragons?
Yeah, I think a lot of that would either come down to its method of visualization. Bearded dragons use binocularity, which it's not like horse tunnel vision, but it's pretty locked on, straightforward vision. And as we can see with the smooth tracking, their head was moving almost at the exact same pathway, which is a very high and pretty well-claimed evidence for binocularity. But more importantly, that can kind of be rooted down to the phylogeny itself with the rods and the cones in the eye. I would say that cones, while they can be able to perceive clarity, the actual image clarity, and also the color itself, that would be helpful for the distance, while the rods, on the other hand, would be more applicable and more photoreceptors to light itself, which can be more contributed to the head alignment. So to say that it might be different would have to be something that is compared and contrasted from species to species or from families like Agamides to others as well.
Still have time for more questions if there are.
Yes.
In terms of your measurement of error, it seems like that's going to have really be affected by how close the lizard is to the bait.
Mm-hmm.
And were there differences in that, say, from when they've got one eye covered or not, or it seems like there's a kind of an experimental issue there, or were they all pretty much at the same distance?
I mean, what my videos kind of showed is that they were a little bit further away and then they slowly start to creep their way in through their videos. And actually, I think Helios's uncovered data is actually really important. So they're a little bit at a distance, but once they lock on, and especially when they start to narrow closer and closer to their object and their prey, they're actually having more perfect alignment, which that's pretty helpful data. But in terms of individual eyes, I would say their ability to slowly move in closer and closer, like we saw from the single eye video of Helios, while they do narrow in, their assessment to consistently go after their bait is heavily hindered. And I think that would definitely have an impact of their distance compared to their alignment perfectly with the bait in front of them.
Do you ever think about just measuring the angular deflection as opposed to the distance from the center line?
I haven't thought specifically of that, but the reason we wanted to do more of a line assessment is especially calculate that kind of error between the lizard and also the bait as those 3 points move back and forth. While it isn't a perfect, exact curved angle, we were kind of measuring what that distance was. And I think that was pretty complimentary and it answered a lot of that angular question too, with the software we use in MATLAB.
Why did you choose to hold the string with the bait rather than having it in a fixed position? Because it seemed like you were moving the bait away from the animal while he was trying to get it.
Yeah, I mean, with the moving object, what we really wanted to understand with the alignment, and I actually was able to do relatively still bait experiments where I was able to kind of hold it still. And in fact, that was kind of the results I used and experiments and methods I used for the distance where I held the bait still. and try to minimize any movement. But in terms of figuring out their alignment, especially if there's a little tiny cricket that is trying to move away, their alignment is really helpful to understand those sine waves and those oscillations as it moves back and forth. And I wouldn't say that any kind of, I would say, patterning or adjustments they made to the pathway was impacted. I think they had nearly similar results, and they didn't progressively increase their accuracy as they got used to the experiment, which I thought was really interesting, but also helpful to understand the broad spectrum of their head alignment and their ambush predator style.
Okay, let's thank Nima for his great talk.